My name is Felix, and welcome to my capstone presentation, in vivo, the first on-demand use of CRISPR. In 2024, an infant named KJ was born with a rare genetic disorder called CPS1 deficiency. This disease affects roughly 1 in 1,300,000 and has an infant mortality rate of roughly 50%. Following only 6 months after his birth, a team of doctors was able to create a patient-specific gene therapy in response, which they called K-APE, and this treatment was not only successful, but it was the first ever personalized in vivo CRISPR gene therapy treatment. First off, what is CPS1? Well, CPS1 is an autosomal recessive disease in which a single nucleotide substitution results in two truncating codons, interrupting the urea cycle. What does that mean? Well, an autosomal recessive disorder is caused when an individual inherits two mutant copies of an allele. Alleles are basically small subsections of DNA, which, depending on the gene, you inherit one from each parent, and together they determine the expression of a specific gene within an individual. In the case of CPS1, this means that KJ inherited one mutated copy from both parents, and that's what caused him to have this disorder. The specific mutation behind CPS1 is a nucleotide substitution. Basically, you can think of DNA as kind of a big book, and it essentially it tells cells what to do whenever cells read it. And cells can read words. You can think of it as codons, essentially, and these codons are made up of letters. Whenever mutations happen, these letters can kind of get jumbled. Like, for example, maybe a cell wants to determine how much it should grow within a specific phase or cycle, and because of a mutation, instead of reading the word fast, it reads the word fart or something like that because the S got switched for an R, and it's confused. And cells can't really think or determine errors like that, so they just try to stick to whatever the DNA says, and they do their best, and that can have unintended consequences sometimes. In the case of CPS1, the substitution basically disrupts the urea cycle. And what that means is that us as humans basically produce a toxic substance called ammonia within us whenever we metabolize proteins. And usually our body deals with the substance through the urea cycle. It breaks down the ammonia into urea, and then we excrete it through our urine eventually. But in individuals with CPS1, this cycle is disrupted, and instead what they have is a buildup of ammonia, which is often fatal because ammonia is toxic. Now, how is this disorder treated? Well, the team that came up with a solution created a customized lipid nanoparticle delivered base editing therapy, and they called it K-APE. This is short for Kjagerin Ebengsemeron and Ebengsemeron, which are both mouthfuls and are the respective names of the guide RNA and the base editor mRNA, which were used in therapy. Their process for this treatment began through isolated testing. Basically, what they did was they took the DNA segments that caused CPS1 and they induced it within lab-grown liver cells, and that kind of gave them a model playground to be able to test affected cells without being at risk of harming or having unintended effects on KJ, right? The testing eventually led them to discover that the adenine base editor abemcemeron and the guide protein Kjagerin were the most efficient methods of DNA corrective therapy. And this therapy is essentially a modified version of a treatment called CRISPR, which is a system which we've discussed in class, but basically it's a system that targets specific DNA sections using a guide RNA, and then uses a protein called Cas9 to essentially cut those sections of RNA. But in the case of this treatment specifically, rather than using Cas9, they used a abemcemeron, and instead of cutting into the DNA segment, they replaced it to essentially correct the single codon substitution, which caused the CPS1 mutation. Following testing, it was identified that Ka resulted in a 42% whole liver corrective editing and showed no clinical signs of toxic effects. Why is this important? Well, an article by the Associated Press states this as the first step towards the use of gene editing therapies for a wide variety of rare genetic disorders in which there are no definitive medical treatments, and The Guardian lists this as a milestone resulting from years and years of progress in editing. The people that came up with this themselves said that this could lead to a future in which the deployment of patient-specific gene editing therapies will become routine for many genetic diseases. Basically, CRISPR is a technology which we've had for years and years, but has never been used in a case-by-case basis like this. And this is a big stepping stone because it eventually could lead to a future where realistically we might be able to do this for every genetic disorder and essentially come up with a future where genetic disorders are no more because of the technology that we have to correct them. Well, this is my bibliography. Thank you for coming to my TED Talk, and first four sources and the last three I used for images. But thank you for listening, and I hope you learned something.